You know, one of the reasons that these guys are phosphorylated also is because acyl phosphates and other phosphate-derived groups in these molecules can be high energy, and so we can use them on an enzyme to drive ATP synthesis. So you're seeing, we kind of went a little bit backwards here, we're seeing in the second half of the pathway how ATP is getting made in 2 steps, but the initial parts of the pathway have ATP getting invested. So we need to use up an ATP to phosphorylate the sugar, and then we use up another one to phosphorylate the sugar again. So steps 1 and 3 are phosphorylations. Right? These are kind of like the easy steps in glycolysis to think about, because they're just ATP and sugar and ADP and then different sugar, right? And so they're all like, you can think about it directly in terms of the way we already talked about hexokinase, right? ATP, glucose, glucose 6-phosphate, ADP. What's the nucleophile, right? What's the electrophilic center? What's the nature of the, if you're attacking on a carbon, it's going from trigonal to tetrahedral. Principles of enzymes that they're all applying they're all applying to these 4 reactions that involve ATP. Either using up ATP to phosphorylate a sugar, or taking a sugar that has 1 or 2 phosphates on it and making ATP from ADP. But either way, it's a phosphoryl transfer rate. Notice that we're investing 2, but we made 4. Right? There were 2 steps in here. There's 2 steps here, and there's 2 steps here. But here it's 2 times 2, because you split the 6-carbon glucose into 2 3-carbon compounds. Right? So between all these ATP steps, right, we have glucose 6-phosphate, and then we have a rearrangement step to make fructose 6-phosphate for reasons that will become clear in a few minutes or soon. Phosphorylate this guy again, get a bisphosphorylated compound, and then we break it. Right? That's what aldolase is doing here, cleaving the 6-carbon to 2 3-carbon sugars. Okay? So this is generating glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And the next step after that is not really a step in terms of a new metabolite, but it's to say, okay, we got these breakdown products, let's interconvert them. So there's an enzyme, this isomerase, this triose phosphate isomerase, which is step 5, is taking these 2 aldolase breakdown products, interconverting them. And what that means is that if this guy is the one that goes forward, which it is, then this dihydroxyacetone phosphate, if this goes forward into the next step by virtue of this enzyme, glyceraldehyde 3-phosphate dehydrogenase, then this equilibrium is shifted so that this guy goes to this, and then this enzyme makes more of that, and then this guy shifts over. So the carbons from glucose initially go to this 3-carbon dihydroxyacetone phosphate as one of the 2 3-carbon compounds after this glucose is split in half. Only one of those goes forward, but the other one does not get thrown away. It just gets re-equilibrated by this enzyme fast, because it's a catalytic reaction, into more of the guy that can go forward, more of the guy that can go forward. So it all gets used. Right? So then we're here, and now we've got the 2, right? Because this guy turned into this guy by virtue of that enzyme. So now we've got 2. And now we've got this part of the pathway by which where the invested energy in these compounds can be used to drive ATP synthesis in 2 steps. Right? These steps here are necessary interconversions to get from here to here, but they're very interesting enzymes, mutase and enolase. And then finally we've got this guy, which is a dehydrogenase running a redox reaction. And this is where glycolysis is making some NADH. Right? So in aerobic cells that retain glycolysis, this NADH is going to be usable to make ATP by oxidative phosphorylation. Okay? Meaning, we're in the cytoplasm here. This NADH has got a journey to do, because this guy's got to get, or in some way, its equivalents, its reducing equivalents, its electrons, have to get to the inner mitochondrial membrane where they can participate in the process of generating ATP. Right? So this is very important. This is the redox part. And so NAD is making, and there's 2 NADHs getting made. So those 2 NADHs alone will generate more ATP, or can generate more ATP, depending on the cell type, than the 2 that are made on the enzyme, the net of 2 that are made on the enzyme, 4 minus the 2 invested in the first part. Any questions about the big picture so far? So that's an overview. So we're going to look at some of the individual steps. Yeah.
I guess maybe this, I'm not sure if this is a great question, but is the big picture to form 2 pyruvate or to form NADH to go on to form?
Both. Pyruvate you need will go on to in the pyruvate dehydrogenase complex to make acetyl-CoA, which is a 2-carbon compound linked to coenzyme A, which we mentioned. And then that will enter the TCA cycle, and a huge amount of NADH will get generated there. So pyruvate is not so important because it's of itself, but because it enters the TCA cycle later, it gets further burned. In terms of glucose to pyruvate is only burning, so to speak, a very small amount of the energy that's actually embedded in the glucose molecule. Yeah. Other question? See in front? No? Okay, so fates of pyruvate. Right? Glucose goes to pyruvate. This is another big picture slide to kind of give you some more context. We're doing this now, right? But what is pyruvate going to do after that? Right? Well, it depends on whether things are aerobic or anaerobic. Because in these cells that are anaerobic, pyruvate is going to go to lactate. Right? Or if you're racing, you're exercising, and you start to feel the buildup of that pain in your muscle, like the acid pain in your muscle, like you can't run any farther. Not your breath necessarily, but your muscles hurt. Right? That's lactic acid. That's this guy. Because you were exercising so much that your oxygen demand was so high that your lungs could not supply enough O2 through hemoglobin to deliver to the muscles. Right? And so you had no O2 to deliver your electrons to. So your NADH in glycolysis couldn't go to oxygen because your body couldn't get it there. And so what's going to happen then? The glycolysis is going to go through. There's going to be NADH made. It's going to make pyruvate, but it doesn't go farther. So it goes into something else, and that's this. And that's called a fermentation reaction. Right? Other organisms also do fermentation reactions. Yeast does fermentation reactions in the absence of oxygen. And of course, the product of yeast fermentation is not lactate, but ethanol. Right? And here it is here. So this is anaerobic, but it depends on the organism. Where are you here? Here are your animal cells, and here are your yeast. So pyruvate then could also go straight ahead, aerobic, release CO2. This was 3 carbons. It's releasing 1 carbon. Now there's 2 molecules of a 2-carbon compound with the CoA cofactor we talked before, which is thioester. Remember the thioester, which is another high energy bond. This guy shoots into the TCA cycle, citric acid cycle, or Krebs cycle. And the ultimate products of this are the complete oxidation to CO2 and the generation of a lot of NADH and FADH2, which goes on to make a lot of ATP. Okay, so that's a picture of a significant portion of the future weeks of the class, where we're going here. Okay, so a lot of this was big picture. So what we're going to do now is we're going to get into talking about the details. Right. So we're just going to talk about every enzyme and see how it goes. And the first enzyme we've already talked about. We don't need to spend a lot more time on hexokinase because we know that it uses the high energy phosphate linkage in ATP to phosphorylate glucose at the 6th position. Right? Depending on the cell type, hexokinase can sometimes phosphorylate other hexose sugars, and sometimes that's an important part of metabolism in other cells. I mean, cells, when they're growing in nature, or even when you grow them in the lab, you don't just give them glucose, you give them a bunch of other sugars too. So we'll talk on Monday about how those other sugars can be used in glycolysis by feeding in somehow. Right? And so this broader specificity can be part of that. Soluble and cytosolic uses magnesium ATP. So in the cell, most of the ATP, almost all the ATP, is bound up with magnesium ions. And magnesium ions are so important to the cell that we even have them in the standard state expression. You might remember 1 millimolar magnesium was in there. So every glycolytic enzyme uses magnesium. Now, some of them will actually use magnesium directly in the catalytic reactions, like magnesium as a Lewis acid, and we'll see that when we get to enolase in step 9. We'll see why we need a magnesium ion right in the active site. But for many of the glycolytic enzymes in these 10 steps, they don't need the magnesium for the catalysis, but they need it for the binding. You neutralize the charge on that phosphate, right? Because all these compounds are sugar, phosphorylated sugars, sometimes two phosphates on the same small 3-carbon sugar. So there's a lot of negative electron density on these compounds. So it's not surprising that they need magnesium, and sometimes the magnesium accompanies the association with the phosphate, accompanies the ligand, the phosphorylated sugar, onto the next enzyme in the pathway. So you might have magnesium bridging the phosphate to maybe an asp on the enzyme or something like that. So the magnesium becomes important for binding the substrate rather than